Okay, for this week's Rhabdomyces Research Roundup, we're joined by Justin Goodrich from the University of Edinburgh. We're going to be talking about a paper he's had recently published in PLoS Genetics, and the title is Kicking Against the PCRs, a Domesticated Transposase Antagonises Silencing Mediated by Polycomb group proteins and is an accessory component of Polycomb Repressive Complex 2. So this fits with the fact that all these titles seem to be very long these days, but I managed to get through them somehow. So good afternoon, Justin. Thanks for doing this. So first of all, let me ask you to give a brief bit of background and then an overview of the of this paper, if you could.
Sure. Thanks, Geraint. So this work has been a very enjoyable collaboration, and it came out of a European grant for the Plant Genomics Network, GARNet, and I was on that with Franziska Turk from the Max Planck Institute in Cologne. And both of us are interested in control of development by Polycomb group genes. And independently, we both did genetic screens for novel genes that might oppose silencing by Polycomb group genes, and we found out in one of the meetings that we'd identified the same gene independently, and this gene's called ALP1 for antagonist of like heterochromatin protein 1. And so that's led to a very nice collaboration, whereas if we hadn't been in that network, we probably would have ended up competing on it. And we're continuing to collaborate. And it's also been, we've been helped a lot by Yuri Rapsilber, who has a proteomics facility in the Wellcome Trust Institute next door to us in Edinburgh. So I guess the take-home message perhaps from the paper could be summarised as it backs up the old adage that it's good to keep your enemies close to you.
Right.
And it's essentially an example of how evolution can make a friend out of an enemy in plants. The plants have sort of very large, unruly genomes which are full of transposable elements. And basically these are parasites. They exist to replicate, and they can replicate faster than their host can remove them. And in general, that replication is deleterious to the hosts. And so there's been a kind of, if you like, evolutionary arms race between the plant hosts and their transposons. And in particular, plant hosts have evolved ways in which they can inactivate and prevent the transposons from moving around. And that's mostly through DNA methylation, inactivating expression of genes that encode for transposases. But there's good evidence that there's also a role for Polycomb group genes and histone methylation, particularly in more basal plants like algae. Now, there are a few instances where transposons can serve useful purposes. For example, when they land in a gene, they can sometimes affect its activity in a beneficial way, and there's a nice example of that from Kathy Martin's work at the John Innes Institute, where she's shown that production of anthocyanins in oranges during cold is a result of a transposon insertion into a gene that promotes anthocyanin production. There are also cases where the actual machinery the transposons encode to get them to jump around has become domesticated and has acquired a new function that's useful to the host. And one of the famous examples of that is actually in the immune system, in VDJ recombination. So in order to produce our diversity of immune responses, there's a protein called RAG1, which basically is a modified transposase, isn't involved in moving gene segments around the genome. So there are a few examples, but not many, where it's known what these domesticated transposase proteins do. And essentially what we found was that the ALP1 protein, which we identified through a role in basically opposing silencing by Polycomb group proteins. So we identified it genetically as a suppressor, Polycomb mutants. And the basis of that is that it acts in a antagonistic fashion to activate genes, whereas Polycomb usually exists to silence genes. So the surprise was when we identified this gene, we thought that it would encode a typical chromatin protein, particularly of a group called trithorax, which are well known for modifying chromatin and activating targets of Polycomb. And in fact, we found that it was a protein related to a transposase, so a protein encoded by a type of transposon called harbinger in animals, or PON or PIF in plants. They're very numerous, this class, in rice, and they've been domesticated in vertebrates, so we all have a gene that's derived from a harbinger transposase, although it's not known what that does. So we found that the ALK1 protein actually encoded a protein that was clearly similar to part of the transposition machinery, a protein which initially is involved in cutting DNA and helping transposon basically jump out and insert somewhere else in the genome. So really that was our first surprise, and initially it was slightly disappointing, but then we had this collaboration with Yuri Rapsilber, and we tagged ALK1 with a GFP tag, and we were then able to immunoprecipitate ALK1 and see what it's doing in the cells. And to our surprise, when we did that, we found that ALK1 was a component not of an activating complex, but it actually purified with something called PRC2, Polycomb Repressive Complex 2, and that's part of the core machinery of Polycomb. It's actually an enzyme complex which makes a modification to histones. It methylates one of the residues, lysine 27, on the H3 tail. So it seems as if this transposase has become part of the Polycomb machinery, and it seems in some way to be inhibiting its activity. It looks from our phylogenetic analysis like this is quite an ancient gene in plants, but that in flowering plants it somehow acquired this new function in antagonizing Polycomb repression. And so that, I guess, was essentially the finding. And I suppose the other aspect is that we think that perhaps initially this arose as a way of transposons maybe fighting back against proteins that normally inhibit them. And the host, in other words, plants, are now using this as a way of regulating Polycomb activity. So that's really the paper in a long nutshell.
Yeah, no, absolutely. No, it's a good bit of background to it, certainly, and broadening out the subject. So you say that it has activity in flowering plants, but do you have any evidence, or are you going to look in future to see whether these harbinger-type transposase elements have similar activity in monocots, maybe, or in other systems, or do you only have evidence that they're active in angiosperms and flowering plants?
It's a good question, yes. It's something that we would like to do. So one thing we've been doing in the lab recently is using Marcantia, which is a very basal lamp plant, as a model system to look at some other genes. And we can find out one going right back to one of the most basal lamp plant groups. Marcantia is a liverwort. So at some time in the future, it might be very interesting to knock it out there. It would also be interesting to knock it out in cereals, because particularly in rice, harbinger class transposases have been very active, and PIF and PON related elements are incredibly numerous in the genome. So there's been a lot of transposition. So it would be quite an interesting sort of plant in which to look at ALK1, whereas in Arabidopsis, transposons are generally somewhat less active.
Yeah, so I think you identified there are specific mutations within the transposase gene which prevent it having transposase activity. So do you see, or have you looked for similar type of mutations or alterations in other transposases, either in Arabidopsis or in rice or other systems? Because that might give you a clue that they have similar activity, or they've been similarly domesticated in some way. I don't know.
Yeah, good question. So in angiosperms, there's a second clade of proteins that are very closely related to ALK1. And they've also acquired similar mutations that have inactivated this catalytic triad.
Okay.
So the suggestion might be that there's redundancy between those, but in fact, when we knocked them out or combined them with ALK1, we saw no interactions. So there's something very closely related in plants that currently we have no idea what it's doing, or at least it's not in that pathway. In earlier plants, so in more basal plants, gymnosperms, etc., you can find out one, but it's not got those mutations. So it might be interesting to see if that can catalyse transposition. And there are systems that have been set up by Sue Wesler and others, which one can test for that with. So again, that's something that could be of interest.
Yeah, okay. So just final question. So this ALK1, are you continuing to work with ALK1 to look at the mechanism of how it interacts with the PCR complex? Is that part of the plans for the future?
Indeed, yes. So we're continuing to collaborate with Francisca. We've identified another gene in that pathway, and unfortunately that's also at the protein level, its structure's uninformative, but it seems to be in the complex. So we're looking at that. Francisca has expressed some of the PRC2 members. So one thing that we'd like to do is test in vitro whether there's an effect on the activity. And there are also groups in Edinburgh that have got expertise at that. So that's one area that we may try to collaborate in future. And we're also interested in the idea that this gene may have a role in normal plants, in other words, ones that aren't mutants, in perhaps overcoming kind of polycomb repression during various stresses or disease attack. So we're looking at that as well.
Okay, excellent. All right, well, we'll look out for those papers over the coming years, certainly. All right, so thank you very much, Justin, for explaining this, and I hope you have a good Christmas.
Thank you very much, Geraint. Same to you.
Thank you.